It's very nice to have the opportunity to participate in this symposium. As Tom indicated, the issue that I would like to address, coming again from an interdisciplinary approach, divergent, but I hope in the end related to what we've just heard, is I guess what I would call biologic and potentially microbial markers to assess environmental encounters and how that shapes susceptibility, particularly for chronic inflammatory disease. These are the points. I'm looking for a pointer to point about. I guess I could use this. Does this work? You know, sometimes the oh, here we go. Oh, okay. I'll save you my red laser story in case we have questions. So what I'll be telling you about are 4 points. One of them is the issue about defining microbial neighborhoods in the gut, and secondly, the diversity of the microbial community in the gut, and how that's actually affected by environment and potentially is biologically important. And then I'll turn to actually evidence in mouse systems that indeed is, and a surprising way that the immune system has become entrained by the microbiota to affect the susceptibility for chronic inflammatory disease using a very conserved sensing system for metabolic stress. So first, your intestines. Of course, intestines is an enormous absorptive epithelial surface, and if one imagines, even this is the inside where your lamina propria would be located, but on the outside, you actually have a pretty good window on what's going on in the mucosa, because there's, first of all, a biofilm of microbiota that live there, which is determined largely by what you eat, and also by how it was formed in the first place. And secondly, by the secretory products of the local host cells, which are responding to that local microbial community. And so we became interested in how we could, people have studied this really for decades, but what people have used to analyze that biology has always been difficult because, for example, if you use fecal samples, you're really looking at the food and the fecal column moving through. You're not really observing what are the microbiota residing in that local biologic environment. And secondly, the readout of the host response has generally been very limited to just certain products that people have, or cell types that people have found of interest. So I was listening with a lot of interest to Dr. Prentice's comment about the question about what sort of markers one would use and the possibility of not trying to be smart, but instead turning to higher dimension analytes to allow the biologic system to teach us. And so what we did is we just made little squirts of the mucosal surface, collected these specimens, divided it into a cellular pellet, a particulate pellet, and the soluble proteins, and then analyzed them using high throughput methods. So we've actually analyzed about 600 individuals using this approach. People undergoing just a screening colonoscopy, at least in large part, where you make little squirts in the endoscopist of just a few mils of fluid from a surface about the surface area of between a dime and a quarter. And then you collect these materials and you analyze them. And the first thing we learned is if you analyze the protein that you obtain, 3/4 of it is of human origin. So even though it's on the surface, most of it is host, not microbial, although a certain amount is a different of microbial origin. And then if we analyze the microbiota, they turn out to have, as people observed by other methods, more than 1,000 different microbial features in them, which is what people are coming to learn as the complexity of the microbiota in the gut. And so when one just simply clusters this data, one finds that it does bin out into different, into its own sort of organization. And we were delighted to find, and this is looking at MALDI-MS, And these are just a small example of samples. And what we observed is that if you just allowed random assorting of the data, they first assorted by the location in the gut where the samples were obtained. And when we did a more formal analysis of that, we found that the sigmoid intestine, the rectum, most distal part of the colon, segregated out as being a distinct site from the proximal intestine, from proximal colon. And so these, just by this simple analysis, identified two biologic regions of the intestine. We also looked at each one of these samples for measuring the abundance of microbial features, molecular features, from the 1,000 or more different microbial phylotypes, which one would identify from each specimen. And so we put those into a large regression analysis and just asked, were any proteins correlating positively or negatively with any of the microbial phylotypes? And this is the data. Among the 50,000 features, there were a few hundred which did positively correlate, and in the microbial features, about 50. And as you can see, it actually sorts into about 5 different regions, 5 different groupings, which are identified by this consortium of microbiota and the proteins that are around where they are around. And we've actually found that these two are present mainly in the sigmoid and the rectum, and the other 3 are more likely to appear in the proximal intestine. So my point here, and I just want to give you a flavor of this, and I guess maybe to show that even immunologists do statistical quantitative analysis, that one could actually use this sort of approach to identify microbial biologic communities in the gut. And that this, we hope, will be the beginning of a way of rationalizing these, the functional features and the disease states or normal states by reducing this very complex data sets to useful biomarkers, which also hopefully are biologically informative. We wanted to use this approach and others to get to this epidemiologic problem. Since World War II, the end of World War II, the incidence of many chronic inflammatory diseases, and I'm just showing you the data from ulcerative colitis and Crohn's disease, has increased more than 20-fold. And so that indicates that that in a group of individuals where there hasn't been a genetic flux in that population, there's something about the environmental conditions which have greatly changed the penetrance of disease. And if we can understand this, that becomes important because there's been a 20-fold change in incidence in a group of people whose genetic risks haven't altered. You might say that 95% of the disease occurrence is really occurring because of environmental factors, where if you can get a handle on them, then you could do something about it, which I know is really a major focus of this center. In Shanghai, in the last 10 years, the incidence of Crohn's disease has increased 10-fold, which reflects the same issue that adopting an urban Western lifestyle is bringing out susceptibility traits. Now, when one looks at the microbial features in the gut, you have this problem. There's more than 1,000 microbial phylotypes that you could identify. Most organisms are not culturable. Also, those 1,000 are different from one person to the next, so it's an insufferably difficult problem right now, although there's a lot of effort and a toolbox which is developing to analyze this. The overall structure of the microbial community, if you go to the genus and family or order level, is about the same. So here's 2 different individuals, and so you could see that the overall structure is the same. But if you get out to the strains and species of organisms, they're really quite different from one individual to the next. So this is somebody from the south, somebody from the northeast, and this is the difficulty that we have. One cool feature besides lifestyle is the founder effect. When you're born, your gut is empty, and all the bacteria rush in. And it turns out that, of course, that the first bacteria that rush in have an advantage. And the reason why we think that's the case is both in humans and in mice, if you analyze the similarity coefficients of the microbial phylotypes, they are similar in individuals that either are genetically identical, not genetically identical, but had the same environmental traits, or even siblings, but it falls off quite a bit with unrelated pairs. And so it seems that actually the maternal input is quite important, because if you analyze this, the relatedness between fraternal twins or siblings is similar to the relatedness of the kid with the mom, much less so for the dad, presumably because in most families the mom has much more intimate contact. And so there's a founder effect as well as that. That's quite important, because one thinks about the phenotype of disease. We're aware, of course, genetics. We're aware the efforts of identifying ecology. But for moms, they're contributing two other things. One is the mitochondrial genome, and the other is the microbiota. So we developed a system to actually analyze this. There's 2 colonies of mice at UCLA which have been kept apart from a long time, and I won't bother you with the details of how they're created. Both of them are quarantine colonies. They have high complexity, 1,000 phylotypes in them, but they're quite different if you analyze them out, and I'm just showing you representative data about them. And what caught our attention about those colonies is that if you brought into those colonies mice which have genetic traits which make them susceptible to chronic colitis, colitis, it turned out that in the one microbial community, there was no phenotype. The mice were healthy, even though they were carrying horribly scary genetic traits for chronic inflammatory disease, whereas if they had the other set of microbiota, they were penetrant. So this is an example of how the microbiota can affect the disease penetrance. It's also interesting that these mice were much less susceptible to diseases which give you certain sorts of cancer. So our collaborator Robert Chistel, who studies ATM, which is a very penetrant disease trait for lymphoma, mice brought into this colony had very little disease after a year, whereas mice in the regular sort of Jackson lab microbiota were very penetrant. They had lymphoma after 3 months and died. So microbiota make a difference, and remember, that's affected by mom, and it's affected by what you eat. So we decided to analyze immunologically what might be the traits which we could use to link the microbiota to this chronic inflammatory disease. I would say that thinking about what we've heard before, there's already 2 sets of biomarkers, one set of biomarkers is available, that in principle, if you could crack the code, then microbial phylotypes might be useful to analyze a set of traits. Right now, that's, as I indicated, a very unruly problem, but in a few years, I think we'll have a toolbox for that. The second, though, is to ask the immune system to tell us what it saw, and then that gives us perhaps a more simple, or the protein features which I was describing before. And so we decided to look at the immunologic features. And what we found is, whereas hundreds of different immunologic traits were unaffected in these 2 mice, there were a set that were quite chains, plasmacytoid dendritic cells, certain subset of B cells, and invariant NKT cells were all very low in the mice which were non-penetrant, were resistant to these chronic inflammatory diseases, whereas mice with the other sort of microbiota had the usual higher levels of those. So this indicated that we had a set of, you might say, biomarkers for this, and we wondered whether they were biologically linked. And so we analyzed them, because this also went with having high levels of memory-activated CD8 T cells. And what we found was that the reason why they were all going up and down were they were actually secondarily reading out the expansion of a cytolytic T cell population, which was directed in its activity by microbiota. So if we had wild-type mice, or mice carrying regular microbiota, they had high levels of, in this case I'm showing you, B cells, but in mice which had the non-permissive microbiota, these B cells were gone. But if we genetically got rid of the CD8 T cells, these cytolytic T cells, then the B cells came back. And the same thing was true for genetically probing them for the cytolytic activity of the CD8 cells. So we had genetic evidence that the pinhole that the microbial encounter was using to affect the immune system was actually expansion of a cytolytic memory CD8 T cell population. So we wondered, what could that T cell population be targeting? And one nice thing about T cells is that there's only a few types of things that they see. They see basically one of 3 or 4 different proteins which are constantly sampling peptides from the interior of the cell and bringing them out on the surface. And so you could ask some very simple questions. Which of these presenting molecules was being used, and what peptides might it be seeing? And surprisingly, over the last few years, we were able to answer that question. The protein that they were using was a class 1 protein, because it's a CD8 T cell. And we also guessed that among the class 1 molecules, the one that it used is a protein called Qa1 or HLA-E in the mouse. And the reason why is because that really is a protein specialized to sense stress proteins in cells. Either it picks up these hydrophobic peptides, stress peptide from certain microbiota, or a stress protein which is expressed in cells undergoing metabolic challenge. And this is how it works. Normally, there's a host protein called HSP60, which is made and then rapidly imported into the mitochondrion. And in that case, it lives its life being a chaperone protein in the mitochondria. But when cells undergo metabolic stress, through a set of mechanisms, this protein is released from the cell, from the mitochondrion, and is rapidly converted into peptides, which then get loaded onto Qa1 or HLA-E. So in a metabolically non-stressed cell, Qa1 is either not expressed on the surface, or it's expressed using a specialized protein which basically tells the immune system to turn off. But when a cell is metabolically stressed, and remember, mitochondria is quite interesting because it's at the center of energy metabolism and sensing, that this peptide is produced and is now loaded on the surface, which is basically an immunologic signal to show the immune system that there's a cell undergoing metabolic stress. And so we analyzed if this is what was going on in cells carrying these different sorts of microbiota. And that turned out to be the case. You could make a peptide, you could make a analytic protein composed of this protein loaded with a peptide, so you can actually use that to analyze these cells. And when we did so, we found that whereas the levels of them are very low in mice carrying the one sort of microbiota, the ones carrying the RF microbiota had high levels. They had an expansion of that population. And we also found that if we took those cells and put them into a mouse carrying regular microbiota, those cells went out and they actually killed cells carrying Qa1. And here's an example of, here's the cells under regular conditions, and if you transfer these cells, just a few days later, they were quite depleted. So it really is a sensing system, and so here's the idea: microbial phylotypes, either directly or by inducing a stress response in the gut, cause a given antigenic challenge, which expands this invariant type of T cell, which is specialized to see stressed products of a metabolically stressed cell. And then they go in to deplete these different cell types. And so we analyzed this immunologically with regard to a chronic inflammatory disease. And so here's an example of chronic colitis, where you could see this is a regular intestine, and here's an intestine with colitis. You could see it's edematous and expanded. And what we found was indeed in this system, like most chronic inflammatory diseases, both for rheumatoid arthritis or multiple sclerosis or colitis, that this genetic disease activity is actually protected if you use a population of these CD8 T cells. And so we analyzed to see whether these T cells were indeed specific for this type of T cell, which is controlled by microbiota and this ecologic input. And what we found is by using the different cell types which you could reconstitute the system with to probe its specificity, we found that it was indeed dependent on class 1 recognition, and that the T cells had to be express Qa1, be specific for Qa1, and had to be competent for cytolysis, their mechanism of regulation. And I know most of you are not immunologists in the audience, but this is an example of what this looks like. That, for example, here is a sick mouse with a lot of activated cells expressing TNF-alpha, but here's a healthy mouse where you can see that the abundance of cells expressing TNF-alpha is reduced. And if you try and do protection in mice where the T cells are, there's plenty of T cells, but they're genetically unable to carry out the cytolysis mechanism, you see they don't deplete the activated cells, which are causing the inflammatory state. And here's the genetics of it with regard to Qa1, that if you transfer in T cells, which are, again, genetically unable to recognize Qa1, when they're genetically able to detect Qa1, then it causes protection. And here's an example of a edematous colon, and here's a regular colon. And if you do weights, then you can see that it goes down in protection. But if you use T cells from a mouse which is genetically unable to recognize Qa1, they're unable to carry out protection. And this is another example of how it looks like by immunologic methods, where if we look at the abundance of activated CD4 cells, which are the ones which are causing the damage, you could see that there's high levels of them. If you put in protective Qa1 specific T cells, you see it gets rid of most of the activated CD4 cells, which are causing the trouble. But if you put in T cells which are genetically unable to detect Qa1, then they're unable to do the job. So the concept here then is that that diet or environment are creating conditions which either permit the expansion of this population of invariant Qa1 T cells or not, that affect the makeup of the microbiota, which then in turn changes the abundance of certain immune cell types, which you could actually readily detect in the blood using a tetramer assay, and also changes in the cell surface, which I described to you before, in the microbial surface by high-throughput protein analysis. And we think that this is one of the biologically important readouts, endpoints of this host-environment interaction, because these cells, whereas under conditions where you can have the possibility genetically to expand cells which are causing difficulty for chronic inflammatory diseases, that if these cells are present, they'll deplete that population and reduce the penetrance of disease. And so the issues then are exactly how this works in terms of the cell types which are involved in this recognition, and what is the exact environmental input which causes these changes in strategies for therapy. In principle, it should be possible to use the proteomic analysis or its surrogates in the blood, or simply using tetramer analysis, although that wouldn't be so good for the women's health study, but maybe something that would be useful in the blood would be useful for that. And then finally, if this is a biologically useful input, if the problem is that environmentally you're not getting expansion of these cells, then a simple intervention that one could imagine is immunizing individuals with the pertinent peptide, because these peptides should cause this expansion, and that should cause overcome the deficits of lifestyle, which maybe are very difficult to change. And so that's something we're assessing in mice, and will, I hope, if that turns out to be fruitful, to look at that in humans. And so I'll acknowledge the wonderful people in my laboratory, some important colleagues here at UCLA, including Tom Drake, who was very important in forming the proteomic approach to this work, and then our other collaborators. So thank you very much.
I had one, John. How it's easy to see just anatomically how all of that affects inflammatory bowel disease as a process. Does it spread to other parts of the body? So would the more systemic chronic inflammatory diseases be equally or at least some affected?
That's a great point, and it is not misleading, but one gets the sense, because the particular disease state that I'm describing is a state which, of course, affects the intestine itself, that one might imagine that this is all just local biology. But in fact, it's systemic biology. And biologically, I think there's two reasons one can imagine for this. The microbial encounter in the gut then affects immune cell types, which are pelagic, and they travel around the body, and they colonize all the different lymphoid sites in the body. This particular population of T cells are specific for a target which is expressed on any cell type in the body which is undergoing a metabolic challenge. And so that means that, in principle, they'll carry out their same role of sort of mowing the lawn of activated immune cell types or metabolically challenged cell types wherever they are throughout the body. So even though the initial encounter which permitted their formation was in the gut, the immune effect is systemic. And in fact, we've seen that because this very same biology in the same way affects penetrance or not for multiple sclerosis, which of course is a disease which affects the central nervous system, and rheumatoid arthritis, which affects sites throughout the body. And then with regard to cancer penetrance, although we don't know exactly the link with regard to the T cell population, we do know that the microbial effect is causing a change in the penetrance for lymphoma, which again is a systemic disease.
Jonathan, could you be more specific about the nature of the metabolic challenge? What is it in the diet that is causing the increase in inflammatory bowel disease?
Well, first of all, we don't know that. In mice, we know that it's microorganisms more than food that's doing it. I presume that food might be the surrogate, or might be the initial issue, might be the human issue, but then the organisms are getting clustered. So then the question is, well, what are the bacteria doing? One thing we do know is this, that programming all of these changes in terms of the target cell can be recreated by simply activating the NF-kappa B pathway, the classical NF-kappa B pathway. So my guess is that any input which would permit a cell to undergo a predominant NF-kappa B input might be what would be doing the job. Now in the gut, although there's a lot of microorganisms in food going through, the gut and the immune system and the epithelium is biologically organized to be relatively blind to what's going through. It's a lot of things you just don't want to know about. You know, it's food, you want to exchange nutrients and hope that things are okay. But there are some sampling systems, but for example, toll-like receptors are not displayed on the apical surface of epithelium. They're displayed in the basolateral surface, which means it's on the other side of the tight junction. So there won't be sensing of microorganisms that are just that are on the surface, because the epithelium is engineered to try and ignore it, only to ignore bacteria which are misbehaving and going to places that they shouldn't be. So I guess in short, what I'm imagining is that the food or the change in the microbiota, presumably because of food, becomes an issue because of products that they make, which then, at least one issue is it's causing NF-kappa B signaling, and those products have to get to where the cells are, where the responding cells might be, which means they have to have some level of intrusiveness.
John, when you showed the Qa1 T cells, they were expanding in the liver but not the spleen, is that correct?
They're about 20-fold higher levels.
And what about in the intestines? And locally, is there Qa1 expressed in both diseases?
There are high levels in the intestine. We've actually had the pleasure of studying this recently in the human, and using the equivalent tetramers in the human, we found that in the blood they're readily detectable. They're about, on a mean of 50 individuals, they're about 0.5% of lymphocytes, CD8 T cells in the blood are invariant HLA T cells, which is really quite cool. So although they range in levels from different sites, they're actually readily detectable even in the blood, which hopefully gets to what Tom was commenting about, how these are pelagic cells. The other thing that I'd point out, and I find this even more interesting, is that individuals range about 50-fold in the abundance, more than that, in the abundance of these. So in other words, some people might have 2%, and some people might have 0%. And we've called a few back, and we found that that's a stable trait. And so this makes me very hopeful that this is indeed, actually my graduate student who was studying this said, well, that's not good. Look how variable it is. And I said, no, that's great, because humans aren't like, black 6 mice living in a certain colony. They're genetically different. They're each living with a different lifestyle. And so to me, that was extremely promising. This might indeed be reading. It at least gives you hope that it's a trait which might be pertinent to the sort of biology which I think this center is trying to study.
Any other questions? While I'm walking down, I'll ask one then, and that's just that obesity and metabolic syndrome has been clearly associated with inflammation of adipose tissue and liver and others. Are there people pursuing the avenue between what you described and those conditions?
Well, there is one, which is that there's been a fabulously theatrical and just simply beautiful work done by Jeff Gordon's group, report in these last 2 or 3 years. Rachel Lee, I think, has been particularly leading the charge in his group, which have studied both mice and people and have indeed shown that the microbial community living in people that are fat are different than in people that are skinny. And it's actually not so much the microbial phylotypes, but the products of them. They're more in the firmicute family. But what's more interesting is that the organisms that have been selected for people that have a high BMI and are not reporting in the same way as other people what they're eating, that their microbial are sort of fat microbiota too, and their metabolic state is different from the metabolic state of those of lean individuals. And if you put somebody on a diet to shift them over, the microbial community and their products change too. And most beautifully, not most beautifully, but most reductively, if you take mice handled in the same manner, and if you take a bacteria from a fat mouse or a skinny mouse, and you put it into a germ-free mouse, the mouse phenotype goes with that. So that if bacteria from a fat mice will make a germ-free mouse, it'll go up in it'll change its energy. It'll change to a new and fatter equilibrium. So I guess the point is that there's certainly a lot of evidence for the microbial encounter, how this links to the inflammatory part. Right now, I'd say that as beautiful as that work is, that the work that has been reported doesn't yet describe a mechanism. It describes a coexisting states, and so we're quite interested in at least one possibility might be the sensing system.
Thank you very much.